Okay, thanks everybody for coming. My name is Sarah Gignoux-Wolfsohn. I'm a professor over in biology, for those of you who don't know me. And I'm going to be talking a little bit about what my lab works on, which is resilience in the Anthropocene, investigating how marine organisms survive in the face of climate change. And this is exciting for me because this is the first time I've given a seminar where all of the work came out of my current lab. I'm not talking about any previous work. So I haven't practiced this talk before, so hopefully it's good. But excited to hear what you think and excited to share our research with you. So we all know that the Earth is getting warmer and warming more rapidly in some locations rather than others. And along with this warming, we have other downstream effects such as changes in precipitation, changes in oxygen levels, especially in the oceans or waters, and changes in pH, again, especially in the oceans. At the same time, in this era of global change, we have a homogenization of biodiversity on the Earth. We humans are bringing organisms that previously had been separated by physical barriers. We are bringing them together. We are introducing species and creating more homogeneous biodiversity across our entire Earth. And so we see this as a gradual process, but also there are rapid changes that are associated with this. And so some examples of rapid changes include wildfire outbreaks, extreme flooding events. This is actually a picture that my dad took in Philadelphia a few years ago. And so these little gray things are lamp posts, and the water was all the way up almost to the top of the lamp post, right? So these really rapid flooding events. And also infectious disease that can be rapidly introduced to a location and have really large effects. And so my lab asked two main questions. The first is, why do some organisms survive or even thrive in this era of rapid changes while others do not? And then how does the strong selection that comes about from these rapid changes alter both ecosystems and individuals? And so we look at these questions at multiple different levels of biological organization. The first is at the species level. So we can have a community of species, and a rapid change can lead to some species surviving while others are wiped out. We also do a lot of work looking at the microbiome and how some microbes that might be pre-existing on an organism might allow them to survive, while a different microbial makeup might cause them to die. We can also look at the microbiome in another way, where there might be a beneficial microbial community here in green that then, because of a rapid change, everybody ends up changing their microbiome in this plastic way. And then finally, the epigenome can play an important role in helping organisms to adapt. And I actually was trying to fix this animation right beforehand and didn't, but so you're going to have to use your imagination a little bit. But so a pre-existing epigenome can cause some organisms to survive while others die, but also the epigenome, like the microbiome, can be changed. So imagine that right now these black ones turned purple and then everybody's purple because after a rapid change, your epigenome might change. Oh, wait, I did it. Okay, I thought I messed it up. I did it. Anyway, ta-da, everybody's purple. Great. Okay, so I'm going to talk about how we address these questions in two different systems. The first one is corals, hard corals. So I'm going to talk about how a novel disease selects for certain coral species and certain coral microbiomes. And then we'll move over to oysters and talk about how oysters can adapt to climate stressors via both the epigenome and the microbiome. Starting with corals. So Corals are animals. They are the foundation of coral reefs, and they come in a lot of different morphological shapes, as you can see here. We have on the left the elkhorn and the staghorn corals, right, that are these tall, really three-dimensional kinds of corals, and then more bouldering and plating. And together they live together and provide shelter for a lot of other organisms, both fish and invertebrates. And I'm going to be talking today about stony coral tissue loss disease, or SCTLD, which is a disease that rapidly causes corals to die. So the live part of the coral is the part that is colored, right? So this yellow color here and this brown here, that is live coral tissue. The white, making up most of this coral here, is actually its dead skeleton because the tissue came off so quickly that it leaves the bright white skeleton underneath. And so this is a disease that can kill corals over the course of several months, even if sometimes those corals are several hundreds of years old. And it is here we go. Yes. So the disease margin here, this is the bare skeleton, and then this is older skeleton that's being overgrown by algae. So this is a new disease. If you guys think 2014 is new, I think 2014 is new. But so it was first noticed off the coast of Miami in 2014. It then pretty rapidly spread along the Florida Keys, and then subsequently through the Caribbean. So we can see that one started in Florida, and it has gone both east and west throughout the Caribbean, most recently ending up in San Andreas here, and these islands that I don't remember the name of down there. So there's still a little bit of the Caribbean that it hasn't gotten to yet, but it has spread pretty rapidly and devastatingly throughout the Caribbean coral reefs. And even though it's been around since 2014, we still do not know what causes this disease. We know that it is spread through the water, that it is infectious, it is likely caused by a biotic agent like bacteria or virus, but we don't know the cause of it. And we care about this disease in particular because its ecological impacts are unprecedented, even when talking about other extreme coral, hard coral diseases. So it affects over 20 different species of coral. It has a high prevalence. It can kill up to 90% of certain species, and it spreads pretty rapidly, as we saw in that previous the map. And then it has the reason why we know that it's a new disease is that it has a unique pattern of species susceptibility, where there are certain species, such as Eusmilia fastigiata up here, Leandrites, that are highly susceptible, while other species are less susceptible and some species seem to be resistant. So we're basically identifying it by the ecological impacts that it has. So my group started monitoring this disease at Caribou Key in Belize in 2019. The disease had not been documented there in 2019. So we were able to go and do surveys and tag corals and collect samples in June of 2019. And then subsequently, the outbreak started sometime between June 2021 and November 2021. Does anyone have an idea why we might not know exactly the start of it? COVID, yeah. So no one was able to go to the field station that we've been using, which was owned by the Smithsonian, or is owned by the Smithsonian, until after November 2021. And so we knew that the disease started sometime there, but we weren't able to capture it right at the beginning. We were able to go back in May 2022, and we found a reef that was in mid-outbreak We then sampled again and surveyed again in December 2022. Okay, and so I'm going to talk a little bit about the species level effects of SCTLD on these reefs. So first we'll start with some species that we know are resistant to this disease and how their abundance on the y-axis changed over time, right? So on the x-axis is time. We have October 2019, January 2020. No. That should be 2019. No, I guess it's 20. Okay, sorry. October 2019 and January 2020. Yeah, right, the subsequent January. And then when we went back, so then we were estimating that the outbreak occurred sometime in July 2021, and then when we went back in May 2022 and December 2022. And so you can see, and then each one of these dots is a different site. So we have 7 different sites that we survey at these locations. And so you can see that while there is some variation over time, in general, these resistant species did not decrease in abundance over time. And you can see these letters that denote whether, so basically if there are different letters, it means that they're significantly different from each other. If it's the same letter, they're the same. And so you can see that in general, they are the same, not significantly different, and they do not significantly decrease over time. And now we'll move to the susceptible species and how they have changed over time. And so we can see that there's a really different story here. The abundance in general decreases over time for all of these species. They are significantly different for everything. Everything is significantly different between 2019, 2020, and 2022, except for Orbisola annularis, which is still significantly similar, or significantly the same, but in 2022, the December is significantly different from the 2019, 2020. So we see that there is this change in species. There is selection of certain species on the part of the disease, and it is changing the makeup of the reef. I mentioned that we also were able to tag and sample corals in 2019. And so we use these little metal tags. They're like the ones that you hang on pipes here. And we basically stick a nail, we hammer a nail into the dead coral or dead substrate or rock underneath, nearby the coral, and tag it with a tag. And then, tag. And then this allows us to go back and revisit the same corals over time. And because the good thing about studying corals is that they don't move. So sometimes it's hard to find them again, but at least they're not going to swim away like a fish or a copepod. And then we're able to repeatedly sample these corals through time. So this is just showing a spot where we took a little biopsy punch of the coral tissue and took this sample, and that'll come into play in a minute. So focusing on the tagging, the advantage of tagging and going back to the same corals versus just surveying in the data that I showed you before is that we can see the fate of these individual corals, and we can see how quickly we can see whether or not they get the disease, and then we can see how quickly they die of the disease or not. So in 2019, we tagged these 4 corals. They were all healthy. You can see that they all have color. They are not white. In May 2022, we went back, and 3 out of the 4 have disease, right? So you can see this active disease where there's a white skeleton being left behind. This one is the only one that was still healthy in 2022. These 2 are also showing disease, or significant disease sites, significant tissue loss. And then by December 2022, that leftmost coral was completely dead. This one is now showing disease signs. These are still continuing to lose their tissue, but still remaining alive. Okay, sorry. And so this fate tracking allows us to look at these individual corals through time and track their fates. And we can look at the different species and how the individuals that we tagged fared over the course of the disease. So starting with the highest susceptible species that we tagged in Belize, Meandrina meandrites. We tagged 13 in 2019, and when we went back in May 2022, all 13 of them were dead, which is pretty sad. Then looking at the intermediately susceptible species, so Montastria cavernosa, Pseudodiploria strongosa, and Cyndrastria siderea, you can see that there are some individuals that are surviving, right? So we have 10 MCAVs that remained healthy regardless of the disease coming through and killing these other corals, and 5 Pseudodiplorias that are still alive, and 6 Cyndrastria sidereas. Also looking at these, tracking these individual corals allows us to see some corals that started to show disease signs, like here, right, there's this yellow, that then when we went back in December of 2022, were healthy again. And so we see that it's not necessarily a death sentence, even if for most corals it is a death sentence. And then we also can see our resistant species that has low susceptibility to the disease. We tag these mostly as a control. And so you can see that of the 17 that we tagged, by the end, 15 of them were still healthy and 2 of them were showing disease signs. I'm not fully convinced that it's actually stony coral tissue loss disease, but they at least showed some sort of tissue loss. But our ultimate goal here is not just to I guess that is one goal, right? So we can look at this at the level of species and see how some species are selected against, see how some organisms survive, and how the ecosystem changes. But we are also interested in looking at the microbiome. Corals, like all of us, are holobionts, which means that they contain other organisms within them, right? So this is a beautiful illustration done by our very own Christian Flaherty, showing that we have the coral animal. So this is like zoomed in on one little polyp. It's almost like an upside down jellyfish. And then all of the members of the holobiont. So corals actually have these symbiotic algae, Symbiodiniaceae, that live inside their tissues and give them nutrients. They also, though, like we do, have bacteria, archaea, fungi, and viruses that can play either beneficial or negative roles in them. These roles can include antimicrobial production, photosynthesis, their immune support, and also potentially gene transfer, but also they can act as pathogens as well. So our overarching question is, how does the coral holobiont evolve disease resistance? And so we're asking this not just on an evolution of an individual animal, but of the holobiont as a whole. And so we can think about the host immune system and genetics, the symbiont community, the algae, the endosymbiotic algae community, and also the microbiome. And so I'm going to talk a little bit about what we found with respect to the microbiome today. So I mentioned that we take samples, just to give you a little bit more detail about our sampling approach. So we're underwater, we take these leather punches and a hammer and hack off a little piece of the coral. This is zoomed in, right? So you can see the place that the punch took a little biopsy of the coral. It includes the tissue and the skeleton. We then put it in, we use like pill boxes, like what old people take their medicine with. They're extremely useful underwater. So we stick that little sample into our pill box. We write down the number of the pill box. We go up on the boat and transfer that to a tube of fixative, and then bring it back here to our lab for processing. And this is work that's being done by Brooke Sinkowitz, who is a PhD student in my lab. And so we've collected the coral tissue samples. She then extracts the DNA and enriches the microbiome, which basically just selects for microbial DNA. We then sequence it using next generation sequencing. quality control and align those sequences, and perform metagenomic analyses. And so what I'm going to talk about today is taxonomic profiling using some software and biomarker identification, right? So this is one way of looking at it. We also are going to, in the future, look at the function of the genes that are within the microbes associated with it. Okay, so our first goal is just to compare the metagenomes of susceptible and resistant colonies. So we have multiple species that you've seen that we tagged and sampled. Pseudodiploria strigosa is highly susceptible. Montastria cavernosa is a little bit less susceptible, a little more intermediately susceptible. And these were colonies that were tagged in 2019. You can then see the disease progress in 2022, and by 2023, they were dead. So this is an NMDS plot. How many people have looked at an NMDS plot before? Okay, just a few. Okay, so it's basically a way of looking at how related anything is, how similar anything is, right? And so here we're talking about how similar the microbiomes are. So each dot represents the microbiome of a coral. The green dots are Montastria cavernosa, the pink dots are Pseudodiploria strigosa. And and then we have three different health statuses. So we have the diseased corals where we took the biopsy from right on the margin of the disease, and then diseased tissue where we took it from the apparently healthy tissue not right next to the margin. And then we have perfectly healthy corals, right? And so those are our shapes. And so the closer together two dots are, the more similar they are. The further apart they are, the more different they are. And here is our results. So as you can see, the pink are clustering over here. The Pseudodiploria are more similar to each other than the MCAS are. The Montastria are more similar to each other, right? So basically, species is what is the driving factor causing differentiating the microbiomes, and the health status is actually less important in differentiating microbiomes. It's more about species than health status. So this was actually surprising. It was both reassuring that species is important, but surprising that health status was not more important. Oh, sorry, disease tissue, disease margin. Yeah. Okay. So, how am I doing on time? Great. So looking at our intermediately susceptible species, Brooke ran an analysis to differentiate the Montastrias that were susceptible to the disease, meaning those colonies that ultimately got the disease, versus those colonies that were resistant to the disease, so that remained healthy. But these are all samples that were taken from before the disease outbreak, so from 2019. And so if you look at this dividing line, basically anything that is above it is going to be more abundant in the susceptible colonies, and anything below is going to be more abundant in the resistant colonies. And she found that there were a lot more taxa that were more abundant in the resistant colonies than in the susceptible colonies. And so this is really exciting because it suggests that there are 11 taxa that might be beneficial for these corals and providing some sort of maybe antibiotic production or immune support, allowing them to fend off this disease. And so these are what we're considering potential biomarkers of disease resistance. And there's one taxa in particular that I was excited to see, which is Endozoicomonas, that I had previously found associated with health in another species of coral during my PhD. Then she looked at our more susceptible species, Pseudodiploria strigosa, compared the resistant and susceptible individuals, right? So there were still some Pseudodiploria strigosa that did survive the disease. And again, the same, if it's negative, it's more abundant and resistant. If it's positive, it's more abundant and susceptible. And she found that there was only one taxon, Vibrio, which was more abundant in the susceptible, nothing that's more abundant in the resistant. So this suggests that maybe these Pseudodiploria strigosa are lacking beneficial microorganisms that might help them survive the disease, and then that's why this species is more susceptible in general. And Vibrio cause cholera, right? So they're like widespread pathogens across taxa, not just in corals. So in conclusion for this part, we believe that there are species-specific microbiomes that might be contributing to disease resistance. And, So in conclusion, coral species are differentially affected by a CTLD, and differences in susceptibility might be due to species-specific microbiomes, and we identify potential biomarkers of resistance. So this is really exciting to our lab because we also have a project where we are trying to make probiotics or coral Go-Gurt to help corals with their immune health, right? So we are trying to source probiotics from natural coral microbiomes and then grow them up in a lab and use them as probiotics. And we're doing this work in San Andres, Colombia, which is one of the most recent places to get this disease in 2022. Even though it only got there in 2022, we've seen pretty outsized effects of the disease on these reefs. And so we're doing this in partnership with a bunch of different universities and nonprofits in both the US and Colombia, and Valeria is our fearless leader, in an attempt to give Caribbean corals a future. And this is work that's being spearheaded by a postdoc in my lab, Nakia Ulrich, where she is trying to identify important microbes and their functions. So we, similarly to Belize, we tagged 4 different coral species and are tracking them through time. And we have taken microbiome samples at multiple time points. She's using metagenomics, where she's trying to assemble the whole genome from this shotgun metagenomics, and she's gotten a few metagenome assembled genomes, and is trying to identify gene clusters of important functions, right? So potentially we can find microbes that have antibiotic properties or microbes that might give some level of immunity to the coral host. And then so our goal is to identify these, to design primers using it, and then you give this information to our collaborators at UNCW and the Ushijima lab, who are working to separate bacteria collected from these corals, grow them up on plates, and screen them for certain traits that we have identified using the metagenomics. And with the ultimate goal of creating basically a treatment for the disease that we can administer in the field. Okay, so this is the hopeful part of the talk. Yay, we're going to make probiotics. But unfortunately, Caribbean coral reefs are experiencing multiple stressors. So those of you who were in class with me may recognize this slide. In 2023 to 2024, there was the 4th globe-wide coral bleaching event. The water was hotter than it has ever been. And when corals bleach, they turn bright white, but they aren't actually dead. So you see this white, it almost looks like the white skeleton from stony coral tissue loss disease, but the coral animal is still there. It has just lost its symbiotic algae. This is what I meant. I'll go back to the other one. So coral bleaching, basically, right, corals usually have these pink symbionts inside their tissues, and then the photosymbionts are either expelled or they're degraded by the coral. And then sometimes corals can move, the arrow goes both ways. Sometimes they can be healthy to bleached, and then the temperature can cool back down, they can go back to healthy, but other times, often, they end up dying, especially when the water is too warm for too long. So we had started this project just focused on the disease. We did an outbreak sampling in May to December of 2022, and then we did a post-outbreak where we didn't see a lot of disease sampling in January 2024, and then immediately after that, this bleaching event started. And so we actually got a grant to go to Belize several times over the course of the year and continue to monitor and sample these corals through that time. So we took 6 trips over this year down there, or members of our group, to continue to sample these corals through the bleaching event and after, and document which ones died and which ones survived. And our future directions of this work, we are working on it right now. Caroline D'Souza and Brooke are both working on this project to explore trade-offs between disease and bleaching, and see how the coral's microbial communities and their algal symbiont communities can shift over time, whether in an adaptive way or a maladaptive way, and be influenced by these different interacting stressors. And then ultimately, it would be great if we could find signatures of resilience to both bleaching and disease, but we also want to look at trade-offs that are causing potentially, corals that are disease resistant to succumb to bleaching or vice versa. So stay tuned for this. Okay, let me drink some water. Coming back to the East Coast of North America, I'm going to talk to you a little bit about oysters and our work on how oysters adapt to climate stressors via both the epigenome and the microbiome. So the oyster that we have here, all up and down the East Coast, including in Massachusetts, and actually as far south as Panama, is called Chrysostrea virginica. And oysters up here or north of Florida basically, form reefs similar to coral reefs. And these reefs provide multiple ecosystem services and ecosystem services for humans, and just they're ecosystem engineers for other organisms. They are amazing filter feeders, oysters. They can remove excess nutrients. They can remove just sediment, like concentrate sediment. This is, I forget exactly what the time lapse, but I think it's like 6 hours of putting oysters in the tank with muddy water that used to look like this, and that's how clear the water is. They're also habitat formers. They create this habitat for other species similar to corals, and they're economically important, obviously, for both wild harvest and aquaculture. How many people like eating oysters? Just Jared. Gosh. I do too. Okay, so in the coastal ecosystems that oysters live in, there are numerous climate change stressors. Obviously, temperature is a big one, but there's also acidification and deoxygenation. And in fact, in the coastal hypoxic site, there has been a 900% increase in coastal hypoxic sites, so sites with low dissolved oxygen, since 1950. So deoxygenation is kind of an understudied but clearly very important stressor. So there are multiple scales at which hypoxia varies. So there's seasonal hypoxia, where you might have more hypoxia in the summer when everything is very active than in the winter. But there's also diel cycling hypoxia in shallow waters, where the oxygen goes up during the day when plants are photosynthesizing, and down at night when there are no plants photosynthesizing. And hypoxia can be problematic for oysters, especially in warmer waters because warmer water holds less oxygen. So the water is getting warmer. There's also less oxygen because of nutrients, but then the warmer water is also holding less less oxygen. And then simultaneously, for a lot of organisms, as the water is getting warmer, especially for ectotherms, their metabolic rate is actually increasing. So their demand for oxygen is increasing at the same time that the amount of oxygen available to them is decreasing. And so we can say that between warming and oxygen demand, much like in Star Wars with the trash compactor, they are squeezed from both sides. I actually went to a conference where I showed this, and then there was someone else also talking about hypoxia and warming, and they had, is it Darth Maul that has like the double lightsaber? And I was like, okay, wow, there's something weird going on with Star Wars and hypoxia, I think. Okay, so our research is focused on understanding how an early life experience of an oyster to a stressor can affect how they respond to that stressor in a future environment. And this is what we refer to as carryover effects. And so we're looking at this one way that we're looking at this is through the epigenome. So the epigenome, there are multiple layers of the epigenome, right? So it's basically anything that's not genetic, but that alters how genes are expressed, how genes are expressed, or even downstream of that, right? So we can talk about chromosomes or chromatin as an epigenetic mechanism, which is the packaging of the DNA. Histones that the DNA is wrapped around, or DNA methylation, which is the addition of a methyl group to cytosines, and these cytosines are usually next to a guanine. I always forget the actual names. A G. The C's are next to a G, and then there's an addition of a methyl group, and that addition of that methyl group above the genome changes how genes are expressed, basically. So this is work that's being led by Julia McDonough, who's a PhD student in my lab. And we designed this experiment where we exposed oysters to climate change, to our two climate change stressors, which is warming and hypoxia, at multiple times in their life. So we had phase 1, where oysters either experienced warm water, hypoxic water that was both warm and hypoxic, or control water that was just ambient temperature and ambient oxygen. They experienced this for 2 weeks. They then had a rest, and then we did a fully crossed experiment where, some of the warm oysters went into warm again, but some of them went into control conditions, and all the possible combinations for phase 2, which was again 2 weeks. And then at the end of this, we took samples of the oyster tissue for methyl rad, which allows us to look at the DNA methylation, and also 16S sequencing, allowing us to look at the microbiome. And so we were only able to sequence a subset of the oysters for the DNA methylation project. So these are only oysters that either experienced hypoxic water or control water, and then again, hypoxic or control at phase 2. And so we sampled these oysters by taking just a little cut of their mantle tissue. Actually, that's not true. We took the whole oyster here. And then we looked at their DNA methylation using this methyl rad technique. And so our overall question is, how does the timing of this hypoxic stress influence the DNA methylation of the oyster? So here we're just looking at how much methylation there is on CPGs. CPGs are just the C next to a G. When their first exposure is control, or the first exposure is hypoxia, and then control or hypoxia, and then up here On the left, we have the second exposure as control, and on the right, the second exposure is hypoxic. And so we see that for oysters that were first exposed to control conditions and then again exposed to control conditions, they had lower methylation levels over all of their CpGs than oysters that were first exposed to hypoxic conditions and then even though they were second exposed to control conditions. So that first exposure seemed to make a difference regardless that they experienced the same conditions in the second exposure. And then we see a similar pattern for the oysters that were second exposed to hypoxic, those that were first exposed to control that were first exposed to control had lower overall methylation levels than those that were first exposed to hypoxia. And then we wanted to know where in their genome this methylation was occurring. So we divided the genome up into exons, which are the coding regions, and then other non-coding regions, promoter regions, et cetera, and looked at first just how all of the CpGs were arranged in these different regions across all of our oysters, and then how the methylated CpGs were arranged. And so we see that methylation is preferentially occurring in our coding regions, in our exons, because there are more exons that are methylated than just that exist. Or there are more CpGs within exons that are methylated than just CpGs that exist in exons. So this is interesting because it suggests that the methylation is preferentially happening on these coding regions of genes, which might suggest that it's important. Okay, so we then looked at differential methylation of these genes across our different controls. So on the top, we have the first exposure to either control, hypoxia, or the gray ones are not significant, so don't worry about that. And then the shapes denote the second exposure to either control or hypoxic conditions. So looking at the effect of early hypoxia, so our control oysters versus our hypoxia control oysters, we see that there was more methylation in the control oysters. And then also, and then we did all of the pairwise combinations, basically. And so in general, you see that on the top, there are more colored dots than on the bottom. And all of the top comparisons are comparing something to the first exposure hypoxic, second exposure control oysters. And so basically these first exposure hypoxia, second exposure control oysters are the most different from all of the other ones. And then we can look at what each of these dots are. Each of these dots represent a differentially methylated gene, and we can look at what these genes do that are different based on our different treatments. And we see that a lot of them are associated with metabolism, which makes sense if you think about oysters dealing with hypoxic conditions. They're probably going to change their metabolism in order to potentially acclimate or compensate for this deficit that they have. We also see a lot of genes associated with signaling, which may suggest that there's something about the methylation that is then doing downstream signaling that's altering gene expression. And then also, interestingly, cellular and oxidative stress response, which again does make sense with the hypoxia, that you would want to compensate for that oxidative stress. And then finally, transcription, which again is probably about the mechanism of the methylation then leading to differences in gene expression. So, this is exciting. This is the first time we're doing it. We then repeated this experiment last summer and have more fine-scaled samples and more sophisticated ways of looking at DNA methylation that Julia is working on now, as she also writes this up for publication. And so stay tuned for more exciting results. So finally, I want to talk a little bit about the oyster microbiome. Just like corals, oysters have microbes associated with them. Bacteria, archaea, viruses, et cetera. They don't have symbiotic algae. And so we wanted to see how these multiple exposures to hypoxia and warming affect oysters' microbiome as a potential alternate mechanism of oysters adapting to climate change. So we have an NMDS, same thing. Each dot represents an oyster's microbiome. The closer together they are, the more similar. The further apart, the more different. The colors tell you about phase 1, and the shapes tell you about phase 2. But basically, sorry, I should have added the legend. But anyway, basically they cluster by the interaction of phase 1 and phase 2, but we see that there's this clear segregation between the phase 1 hypoxic oysters, regardless of the second exposure to hypoxia, or regardless of the second exposure. And then on the right is the phase 1 normoxic oysters that cluster together regardless of their second exposure. So this is interesting and exciting because it suggests that early, just like with the DNA methylation, that early exposure to hypoxia is altering their microbiome in a way that is not reverting, even if they're then exposed to control conditions after. And so we wanted to drill in a little bit and see what species are driving these differences, because that NMGS just shows you that they are different. It doesn't tell you what is driving the difference. And we found that the main difference was in these spirochetes. And spirochetes are actually nitrogen-fixing bacteria, so they're important in nutrient cycling. And we see that if we just look at the spirochetes in these oysters that were first in normoxic conditions, the color basically tells you what strain of spirochete it is, right? So not knowing anything, you can see, okay, these all look pretty similar, right? They're all this like olive green color. Normoxic oysters all have this olive green spirochete. Oops. The hypoxic oysters had totally different strains of spirochete. So this symbiosis between the olive green spirochete and the oyster seems to have been disrupted by that early exposure to hypoxia, and it doesn't ever go back, even if they're subsequently exposed to normal conditions. And actually, that olive green spirochete is something that I've found to be a common associate of Crossostrea virginica in other locations. So this was from a completely different study where I took oysters from Virginia, not Maryland. They were like different oysters, and every single one of these oysters had that same spirochete, but they were all growing in normal conditions. And so this hypoxic exposure is like disrupting what is potentially a conserved symbiosis between the spirochete and the oyster. So this is very preliminary, but I'm really excited about it. I think that it's interesting to look at the potential for spirochete, the potential role that spirochetes play in helping oysters adapt to the environment, and just understanding the symbiosis better. So to summarize this part of the talk, the early exposure to hypoxia led to more methylation, and we identified several differentially methylated gene regions involved in metabolic processing, signaling, stress response, and transcription. And then early exposure to hypoxia also changed the makeup of the spirochete community in a way that was not reversible. And with that, I would like to thank the members of my lab who worked so hard on this, and our collaborators and funding, and take any questions. Yeah.
But how would that control what, like, you had like phase 1, phase 2? Was that not just shrinking control down from a control from phase 1, phase 2? And then like anything that the control changed into like hypoxia, I forget the other two categories. Does that not just mean like a shift in environment rather than just it maintaining to be a controlled experiment?
Yeah, there were only some of the oysters were control in both. So there were some oysters that were in control conditions the whole time, right? But other oysters only experience control conditions at either phase 1 or phase 2. Yeah. Any other questions? I'll give you extra credit if you ask a question.
Is that a stony coral tissue loss, like still really running rampant? Because that was from like 2022, I think was last data.
Yeah, so in Belize, we don't see a lot of it anymore, but part of that might be that there is the trade-off between bleaching and disease, whereas corals bleach, they seem to get less disease. And so then we were following it through the bleaching and afterwards into recovery with the expectation that the disease would ramp back up, but it hasn't really yet. So in Belize, at least, it doesn't seem to be as big of a problem. I mean, it killed like 70% of the corals. So like part of it might just be that there aren't corals left to get sick. But yeah, it doesn't seem to be as much of an active problem right now. In other locations, like in Colombia, in San Andres, it's still, we see corals popping up with disease signs all the time, but it might be because it only got there in 2022, so it's more recent. We are anticipating that it's going to get to Panama soon. Yeah.
I mean, we focus more on like the Colombia area, but like have you seen or like heard of massive outbreaks like you're seeing down there in like Australia and like other regions of like giant corals?
Yeah, no. So it seems, hopefully, like it only affects Caribbean corals. People have done some experiments with Pacific corals, and it seems like they're more tolerant of it, although I think we need to do more experiments to really show that. There is a fear that once it gets to Panama, it'll pass through the Panama Canal and then be on the other side. And so we don't really know what's going to happen, but hopefully they're not as susceptible to it.
Is it entirely new disease for the coral, or is it different strand of an outward or existing?
We don't know what causes it, so it's hard to say, because it could be something, it could be a different strain of an already existing coral. Absolutely. We haven't been able to isolate a pathogen. Yeah.
How did you keep track of the tags, the locations for the coral? Because it looked like just kind of like sit next to one.
Yeah.
Swim.
It's hard. So what we do is when we tag them, we set out a transect line, so like basically like a long string that has numbers on it, right? And so then we make a map, so we're like swimming along, you say, okay, at meter 1.5, if you turn to the right and go 2.5 meters, there should be a coral with a tag there. And then the tags get overgrown with like everything. And so we also take pictures, and then we print the pictures out on underwater paper. So then you're like underwater, you're flipping through a book and being like, is that coral the same coral that, okay, yeah, it is. And then usually you'll be like, oh, the tags should be right here. And then you kind of like clean some algae off and then hopefully find the tag. Low tech, but it works.
Do we anticipate that if SCTLD is spread from the Panama Canal to elsewhere, worst case scenario, will it have the adaptations necessary to spread into perhaps colder climates, maybe?
Yeah, I don't know. I mean, hopefully not because, well, I mean, corals, that's not true, actually. We have corals up here. There are cold water corals here, but they're not reforming. They're not in the same, like, contiguous area, right? So hard corals in general mostly live in warmer waters. Wait, sorry. And if you look at the map, it basically stopped as the water got colder, but also the coral stopped, right? So it spread north initially and then more slowly spread south. But then it hasn't like gone any further. There aren't other organisms that might live north of Martin County that seem to show disease signs. So It could just be that there aren't corals north of there, like the corals are more spread out, they're different species of corals, but hopefully it won't adapt to cold water. Any other questions? No? Okay, cool. Well, thank you guys for listening to me.